Okay, sorry guys, if you hear my daughter in the background screaming, but if UB wants me to teach from home, this is what you're gonna get. Okay, so we were talking about oxaloacetate and it has 4 carbons, so that's why this is called a C4 cycle. With the help of an enzyme called malate oxaloacetate will be converted into malate. And this is kind of an alcohol-like molecule, which will then move onto the bundle sheet cell. So then this is the place where CO2 will be released from malate with a malic enzyme, and we are then left with a pyruvate. So pyruvate will then migrate back into the mesophyll cell and then being converted into phosphoenol pyruvate. So you can see that Calvin cycle is happening in bundle sheet cells, not in the mesophyll cells. So C4 cycle is happening. Calvin cycle in C4 plants is happening not in the mesophyll cell, but in the bundle sheet cell. So C4 plants manage to find a way to kind of store carbon dioxide in the form of malate. So even though you can have low concentrations of CO2 in the atmosphere, there is no danger that Rubisco is accidentally going to fix carbon because, Rubisco is way over here at the beginning of the Calvin cycle, okay? So there is no oxygen right here in the bundle sheet cell. So, this plant, the plants are kind of displaced the cycle into the bundle sheet cell, avoiding the danger of Rubisco accidentally fixing oxygen. So what is going to happen is that you will have a very high CO2 concentration in the bundle sheet cells. And yes, there's some additional energy that's being spent, but it's worth it because then these plants don't produce, glycolate, which is dangerous. These are some of the differences between C3 and C4 plants. And what I would like to point out is that C4 plants can perform photosynthesis even though when stomata are closed because they have those pool of malate. So malate is usually stored in the vacuole. So that's why, C4 plants can photosynthesize much more efficiently. Another thing that C4 plants have is something that is called, I'm going to try not to break my tongue, peripheral reticulum. Okay, I'm not repeating that word twice, but it is like a numerous vesicles or sacs which are located inside the chloroplast. And if you read this text, you will see that botanists think that these sacs can increase the surface area. So that's why the photosynthesis can run more efficiently. So the question really, what we should be asking ourselves is, we have C3 plants which are not so efficient and they don't photosynthesize. But then we have the C4 plants which are more efficient and they photosynthesize way better. So like, why didn't they take over the world? Why isn't like that the majority of plants are C4? Now, one of the reasons Rubisco is so inefficient is that Rubisco, when plants were first evolving, there wasn't a lot of oxygen. So those first cyanobacteria that appear, they evolved in a high CO2 concentration. So only when when the first photosynthetic cyanobacteria appeared, they started pumping oxygen into the atmosphere. So rubisco, as an enzyme, evolved in a high CO2 concentration. Okay. Now, sea plants evolved some 350 million years ago. Now, the CO2 concentrations, they kind of were going up and down throughout the years. And of course, this is, you see the peak in the present day, showing the global warming industrial revolution. But C4 plants evolved only recently. Actually, C4 metabolism evolved at least 14 different times. So it didn't appear once in evolution. So many plants, were trying to establish this C4 metabolism. So C4 plants are kind of new kids on the block. And, they could not take over the world because the world or planet Earth was already taken over, and there were a lot of C3 plants. So that's why, C4 plants didn't kind of diversify rapidly. So, yes, they have a more advanced photosynthetic system, but, they cannot really take over the world, as this squirrel is trying to jump on the roof because, like the block is already taken by somebody else, which are C3 plants. So to sum it up, some of the plants that are famous C4 are pineapple, corn, and maize. And remember, they displaced the Calvin cycle in, like kind of moving it from mesophyll into the bundle sheet cells. And now in the end, we have something that are called CAM plants that have so-called crassulacean acid metabolism. Okay, these are things such as aloe vera, succulent plants, and cacti. So they live in extremely dry environments. And as if you remember, during the day, their stomata are closed because of the very strong sunlight. Their stomata have to be closed during the day to reduce the transpiration. So their stomata are open only during the night. So that is the time when they can fix carbon dioxide. And pretty much CAM metabolism is something between the C3 and C4 metabolism, because right now here you can see they also fix bicarbonate onto phosphoenol pyruvate via this PAP carboxylase. And the oxaloacetate is then converted into malate and then stored into vacuole. And I always joke here with my students, telling them that if they ever get lost in a desert, do not try to drink and eat the cacti in the night, because it's going to be very sour. That's, where malic acid is being stored. So eat the cacti during the day. Okay, so then, from the vacuole, the malic acid will now be released into the chloroplast. And so from malate, of course, CO2 will be released into the Calvin cycle. So what you see here is that the Calvin cycle is happening only in the mesophyll cell. However, what have happened here is that CEM plants, they separate this process. So the fixing of the carbon and the process of Calvin cycle, they separate those processes in time, okay? And C4 plants, they separate these processes in between the mesophyll and the bundle sheet, okay? So, I mean, this is pretty amazing, because this is what enable plants to live in the most extreme environment known on planet Earth, which are, deserts. So that's really kind of amazing.